All right, so in this activity, we are going to measure cellular respiration. And to measure cellular respiration, we're going to construct these little micro respirometers. I've already taken a 1 milliliter syringe and glued a small diameter capillary tube to the end. I can tell that this is airtight because when I pull back on the plunger, it kind of sucks it up a little bit. See? So we need this to be airtight because we're going to be measuring the consumption of oxygen within this chamber. So remember, cellular respiration is a really crucial metabolic pathway where organisms take in oxygen and they release carbon dioxide and water as a byproduct. That's how they break down the sugars in their food. So we want this to be sealed because the whole point is going to be to measure the amount of oxygen that's being burned up or utilized by the organisms that we're going to test in this micro respirometer. So I think we're good. We have a seal here. So I'm just going to start by getting this thing a little bit wet and a little bit soapy. So this is just soapy water with red food coloring. So now the capillary tube is kind of like lubed up. Then I'm going to put a little bit of absorbent cotton inside the barrel of the syringe. So I'm just going to put a tiny little ball in there and then poke it down with the plunger. I'm doing this because I'm going to apply, I need a little bit of KOH inside my micro respirometer. The deal with the KOH is that within cellular respiration, there's a couple of things happening with gases. So while oxygen is being consumed, the gas carbon dioxide is also being created. And if we have one molecule being generated that's a gas and one being taken up that's a gas and converted to a liquid form in this case, we may not see the volume of the gas inside this chamber change at all. So we want to just kind of strip the carbon dioxide that's being generated as a result of cellular respiration. And the KOH will do that for us. So I'm just going to put a drop of KOH down here, and that's going to take care of the carbon dioxide that's generated by the reactions of cellular respiration. But we're going to have living organisms inside this barrel, and they might be harmed by the KOH. So I'm also going to throw a little plug of this non-absorbent cotton in here. It's not going to absorb the KOH. This is just a little plug to protect the organism that we're going to put inside of here and measure its respiration. The organism is a plant. We're going to use radish seeds, and I'm going to put half of a milliliter. So I'm going to put the seeds in here volumetrically. I'm going to put half a milliliter of seeds inside my micro respirometer. So we're just going to dump them in there until we're at 0.7. I need a little bit more. And so this is it. I'm going to put the plunger back at 1 milliliter. Eventually we're going to seal up this side of the chamber and hopefully we'll be able to watch the seeds that are inside of the microrespirometer change the volume of gas inside this chamber as they consume the oxygen that's inside, as they perform cellular respiration. All right, so I've actually created 2 respirometers, and they've been sitting in this water bath for about 10 minutes now. And the idea here is to try to stabilize the temperature, and generally temperature is going to be more stable in the water than it is in the air because of the high specific heat of water. So it's more resistant to temperature change. The reason why we want to minimize temperature change is because remember that we're going to be measuring the change of the volume of the gas inside the microrespirometer. We want that change to be due to cellular respiration rather than a change in atmospheric temperature. So we're trying to stabilize the temperature by running our microrespirometers in a water bath. I also want to point out that the microrespirometer on the right here has live seeds in it. So these guys are germinating, whereas the respirometer on the left has an equal volume of dead seeds. These have gone through the autoclave, and they should not be respiring because they're dead. So we're using this as a control. And if we do see changes in the volume of the gas inside this microrespirometer, we should probably use it to correct the changes that we see over here in our treatment, the microrespirometer that has the live seeds. So I'm going to put this manometer fluid, it's the same red fluid that we originally put inside these capillary tubes. I'm going to put a drop of them inside both, right at the end of both of these capillary tubes. And this is sealing the tubes. Get a little more in here. And I'm just going to pull these down. All right, so I kind of sucked the drops of manometer fluid down the capillary tube a little bit. And now I'm going to put a mark on each of our capillary tubes, showing us the initial position of the bottom of that drop of fluid. And at this point, we're going to wait 5 minutes. So we're going to set a timer for 5 minutes. We'll come back in 5 minutes and we'll mark the position on each of the tubes once again. All right, so this has been going for almost exactly 5 minutes. So I can tell that the respirometer, the manometer fluid is moved in this chamber. So our original dot's up here. I'm going to put our next dot right here. It doesn't look like the control respirometer manometer fluid is moved at all. So there's really no place to put the second dot there. So we do see some movement. The manometer fluid is moving towards the seeds, indicating that the volume of the gas is shrinking. That's a result of cellular respiration occurring in the germinating seeds, but we see that it's not happening in our control, which makes sense because those are not live seeds. So we'll check on this again in 5 more minutes, and we'll mark our manometer fluid position after 10 minutes. Okay, so it's been just about 10 minutes since we put the manometer fluid in the capillary tubes, and I can see that the bottom of the fluid line has shifted once again. So I'm going to put a dot at the bottom This one still hasn't moved. So the water temperature is staying steady at about 26 degrees Celsius, which is good. So we're not seeing any change in volume as a result of a change in temperature. We're seeing a change in volume as a result of cellular respiration occurring here in the respirometer with the living seeds. We're not seeing any change so far in our control. So we will check in on this again in 5 more minutes. So that will be when we make our mark for our 15-minute time interval. Okay, so it's been almost exactly 15 minutes since we put the manometer fluid in the capillary tubes. The temperature of the water bath is holding steady at 26 degrees Celsius. As you can see, the manometer fluid is moving down still. So I'm going to put a mark at the bottom of the manometer fluid for our respirometer containing the living seeds. There's still been no movement in our control, so we're just going to keep that one black dot. There's no reason to do any other marks on that one. So we will check in on this again in 5 minutes, and then we'll record the movement of the manometer fluid. That'll be after 20 minutes total. Okay. So it's been just about 20 minutes since we initially put the manometer fluid into these 2 capillary tubes. So I'm going to mark the distance that the fluid has traveled so we can see that it's still moving in our treatment. And we can see that in the respirometer with the dead seeds, we have no movement. So the temperature of the water bath has not changed, so that partially explains why we're not seeing any movement. That's good, that these have both been under a consistent temperature throughout the entire experiment. So this was our 20-minute reading. We're going to check in on this one more time in 5 minutes. Okay, so it has now been 25 minutes since we first put the manometer fluid in the capillary tube. So I'm going to go ahead and make our final mark. I still see no movement in our control syringe. So we are going to take a picture of both of these setups, and we're going to take a picture of it next to a ruler so that you can actually calculate the rate of respiration that was occurring. So how much gas, oxygen specifically, was being absorbed per minute in our micro respirometer. So thanks for watching. You should be able to complete this activity by examining the photograph that's associated with it.